after the completion of this webinar. You all will be kept on a mute during the webinar. So if you have any questions regarding to the talks, kindly share it in Q&A window, and we will be answering as many questions as many possible during the live after each presentation. If you are on Twitter, kindly tweet about this webinar, tagging our official Twitter handle @Thieme Chemistry and using hashtag Cheminar. I also request you to kindly subscribe to our Cheminar series to keep informed and access all the previous and upcoming webinars, and our newsletter to access our new published content in our journals. Now, I like to give you a brief introduction about the portfolio of chemistry journals and e-products that we have at Thieme. Thieme is a 120 years old publisher. We have quite a legacy in publishing industry. We are publishing organic chemistry journals since last more than 6 decades. We have quite a products which are very well known in the market and amongst the community in organic chemistry. We have a database, SOS, and pharmaceuticals. Then we have a journals like Synthesis, Synlet, Synfax, and the new journals, SynOpen, and Organic Materials. Recently, we have also launched another journal by name Sustainability and Circularity. This is a journal which is aimed to tackle the sustainability issues and shape the interdisciplinary agendas to resolve the global crisis that we face with the increased pollution and other problems in our society. This journal basically is on covers all the topics on sustainability and circular chemistry. So if you are working in this area, the submissions are open. I request you to submit for this. Editor-in-chief for this is Professor Chris from Netherlands, and we also are recruiting reviewers for crowd review. So if you are interested to review for us, kindly share your CV at Sustainability and Circularity Now @thieme.de so that we can include you in our clouds. Now, as most of you are in this webinar are associated with the pharmaceutical industry, I also like to share with you one of the database, which is very good database for pharmaceuticals, people working in the pharmaceutical industry or pharmaceutical chemistry, that is a pharmaceutical substance. This is a database which has a 2,800 APIs and key intermediate information. It has patent, it has a synthetic route, it has information which is very useful for regulatory purpose, and we update it twice a year to include new approved APIs from FDA, and soon we are going to launch new interface for this. If you like to access it, kindly visit our website ps.thieme.com to know more about it. Also, we have a call for papers open for a number of special issues. If you are working in any of these areas, I request you to kindly submit papers for these issues. Now, as we start this seminar is chaired by Professor Joydev Laha. Professor Laha earned his PhD degree in organic chemistry from National Chemical Laboratory in Pune. He followed it with the 5 years postdoctoral stint in synthetic chemistry and medicinal chemistry from North Carolina State University and Mayo College in United States. He also, after completing his postdoctoral process, he was joined drug discovery in neurodegeneration lab in Harvard Medical School, and then in 2011 he moved back to India to start his independent career at National Institute of Pharmaceutical Education Research in Mohali, which is one of the premier leading pharmaceutical institute in India. He currently, his group's focus on organic synthesis for preparation of heterocycles, key starting materials, drug intermediates, and APIs using radical reactions. He has mentored 12 PhD candidates, 2 postdoctoral and 80 master students in his career of 12 years. I now hand over to Professor Laha to start this webinar. To you, Professor Laha.
Yeah, thank you very much, Rohit. Let me share my screen. Is my screen visible?
Yes, sir, it's visual.
All right. Okay. So first of all, I'd like to thank Thieme Chemistry Journal's team, especially Dr. Rohit Bhatia, for organizing this wonderful seminar, and also giving me an opportunity for chairing this event. So, as you know, this seminar, as it shows, is entitled Synthetic Development of Key Intermediates and Active Pharmaceutical Ingredients. So, how this seminar is linked with a special issue in Synthesis Journal, that I'm going to show in the next slide. So this year in 2024, a special issue was published in Synthesis Journal on the topics, the same topic that is linked to this seminar: synthetic development of key intermediates and active pharmaceutical ingredients. And this special issue was guest edited by Professor Steve Zhou in Peking University and I. So we joined together on this special issue, and that was published in this year, sometimes in February. So first of all, I'd like to sincerely thank all the contributors to this special issue. The contributors were from academia and from industry. Am I audible clearly?
Yes.
Okay,
Yes, fine.
all right. So yeah. So as usual, the manuscripts were peer reviewed. And in the event of many submissions, actually, manuscripts were peer-reviewed and many manuscripts were also rejected. And that's because those manuscripts do not meet the novelty and standard of the journal. So that's why we had to reject some of the manuscripts that were submitted in the special issue. So the special issue was related to the pharmaceutical organic synthesis. Pharmaceutical organic synthesis. So in this case, I just wanted to remind you, those who have organic chemistry background. So these pharmaceutical organic synthesis topics is substantially different from routine organic synthesis, in a sense that here the focus is not only to develop chemistry but also to prepare a focused targets like pharmaceuticals. So because of time limit, I cannot go through all these statements which is mentioned here. Yes, I'm trying to do that. So in that case, this pharmaceutical organic synthesis is substantially different, and then there are different parts: medicinal chemistry and process chemistry. So industry has some sorts of compatibility to practice the reactions in industry. So these are some are listed here. They need safer process, easy to handle, use of feedstock chemicals and all. So what I what I'm just trying to say, academic strength we have in terms of we can develop modern chemistry, catalytic chemistry, tandem and domino reactions and all. So these certainly can be, applied in industry, but then there needs to be some demonstration. Okay, and also to mention there are certain weakness from academic environment, academic people, in terms of how to scale up a reaction, how to meet with the cost-effective process, and of course we are not very much expert in cost calculations. So based on this, we thought about what could be the venue of such kind of literature manuscripts. So although there are certain journals available, but then only a few journals are very much dedicated to such kind of organic synthesis papers. Based on that interest, we got involved in this special issue. Okay, now I move to my research interest. Why we became interested to develop this special issue. So as I mentioned earlier, that we have also interest in process chemistry, and you can see from this slide that we are involved in many drug synthesis, and so we follow certain process chemistry rules that could be followed in industry. Based on that, you can see on the left column, we try to develop a novel chemistry, and that we try to apply it in the synthesis of the drugs, like in the reaction of acylation, formylation, amidation. There are many amidation reactions that are known in the academics, but then why they are not used in industry? So that's what one needs to really put much effort to bring it to that level. And also you work on nitro reduction. These days we started. So our research interest is very much focused on process chemistry. And then based on this research interest, we actually got involved with this issue. So I'll just quickly show you one or two more slides on my research interest, how we made a drug sildenafil just using an amidation procedure, not involving any the common coupling reagents. Again, because of shortage of time, I just moved to the another two, three slides. So here I show you how a drug can be prepared telescopically. Means without purifying or isolating the intermediates, and towards the end we can isolate the compounds. So this is very much practiced in industry, and this could make the process very cost effective. And to show you more confidence that we really work on large scale synthesis, try to find out the problems, so based on all these things, we work on process chemistry. And finally, I just wrap up my talk to thank you, the Rohit and other teams of the Thieme, and then these are funding agencies and all. So I just stopped my presentation. Yeah, hello. Yeah, I just stopped my presentation here, and then I will go to the our main event today to listen all the four speakers that are present today. And then the first speaker of the day today is Dr. Luca Laraia. I'm sorry if I'm pronouncing incorrectly. He's currently an associate professor in the Department of Chemistry at Technical University, Denmark. Before joining the faculty position, he had a master's degree from Imperial College London, and then he had a PhD from University of Cambridge in medicinal chemistry and chemical biology, working with Professor David Spring and Ashok Venkataraman. And then he had a postdoctoral experience at Max Planck, working with Professor Herbert Waldman. And then he joined assistant professor in the University of Denmark and then subsequently was promoted to associate professor. So with this, a short introduction, I invite Dr. Luca to, give his presentation.
Yes, thank you very much for the kind introduction. I've just shared my screen and I will share my slides now. So let's see if that. Okay, can you see the normal mode, or is it in presentation mode?
Looks great.
Looks great. Great. Okay, great. Yeah. So thanks a lot for the kind introduction. It's a real pleasure to present sort of our lab's work in this Cheminar. So my talk is entitled "The Next Generation Strategies for the Synthesis of Functional Natural Product Inspired Compounds." So I'll get a little bit into what that means in just a second. But for those who are not familiar with our lab's work, we do quite a lot of different things, of which sort of synthetic chemistry is a part. It's not the only part. So we have so on the sort of top left here, we spend a lot of time making natural product inspired compound libraries, and that is what will be the main focus of my talk today. But the goal, of course, of making such compounds is to apply them in different contexts, and the one that I will focus also on today is to develop tool compounds for sterile transport proteins. So these two kind of boxes will be the focus of today. We do a lot of cell biology, protein biochemistry, and so on, and chemical proteomics, and I won't talk about this and also our work on PROTACs today. That will be for another time. So before I get into what we do, it's worth a little looking at basically what has been done in the field of natural product inspired and derived compound libraries. And of course, one approach is one can take, for example, readily accessible natural products themselves and subject them to different sort of ring expansion, modification, cleavage strategies. And this is sort of known as this complexity to diversity type of approach that is pioneered by the Hergenrother lab and others in the last decade or so. This is good, but you rely on this readily accessible natural products, of which there are not necessarily that many. But in parallel, of course, people have developed diversity-oriented synthesis, which relies on using the power of synthetic chemistry to sort of access both stereochemical and scaffold diversity, and using the sort of build-coupled pair approach pioneered by Stuart Schreiber's lab. And this has also been going on for about 20 years or so. But in this case, there's no requirement for natural product sort of elements in this, although it is often implied. What I've been working on, and both in my postdoc and beyond, is the use of sort of starting from natural products as sort of the guiding elements in library synthesis, but basically either synthesizing natural product scaffolds and then modifying these, or taking so-called natural product fragments and then recombining them in ways that has not been present, that are not found in nature. So these are two sort of complementary approaches developed mainly in the Waldman lab, sort of biology-oriented synthesis, and this slightly newer pseudo-natural product approach, which is kind of what we have also been working quite a lot on. One thing that I want to point out in our work is that actually there's we don't have a favorite in this kind of between these approaches. In fact we quite the opposite. We like to sort of use all of these approaches together basically, and we believe that the real strength lies in sort of combining these rather than sort of being we're favoring one over the rest. And to come back to my title of actually using these in a functional sense, we really ask ourselves, well, which strategy is most conducive to useful biology? So basically one of the main aspects of our lab's research is working on cholesterol transport proteins. And I'm not going to go into depth of the biology here. This is a cheminar, not a bionar, but basically our interest here lies on the fact that there's about 12 or 13 proteins inside the cell that are responsible for trafficking cholesterol between different organelles, such as the ER, lysosome, and trans-Golgi network. And basically, there's a lot of proteins that can do this. So we were interested in understanding how we can sort of decipher the individual activity and function of each of these steroid transport proteins, and basically, and also whether we can hijack this for sort of therapeutic purposes. And there are several challenges when we want to do this. One of them is functional redundancy. So if you use genetic methods to basically knock down or knock out these proteins, typically the cell can recover by overexpressing another protein in the same class. So it's very difficult to really study these proteins using sort of traditional genetics. So of course, as chemists, we like small molecules. We're a medicinal chemistry lab as well. But the challenge here is that there's actually very few known inhibitors of cholesterol transporters, and they're either sort of highly complex natural products, and such as Reterazine B. This has been made actually synthetically, but it's a it's a 50-step total synthesis, very beautiful synthesis by the Reisman lab and others, but of course quite a challenge to access a lot of material. And otherwise, we have these more simple steroidal compounds, but these have very low selectivity. So this is one of the biggest challenges that we have, is how do we selectively inhibit one steroid transporter and not the rest? And the reason why that's such a challenge is that these steroid transporters have a lot of structural similarity in the lipid binding domain. So again, it's very challenging to design ligands that will selectively inhibit one over the rest. So our strategy for designing this sort of builds on these different natural product inspired compound collection strategies. And here we basically use these, so the goal is to use sterile derived primary fragments, such as those shown in dark blue here on the left-hand side of the screen. And then we basically synthetically recombine them with biosynthetically diverse secondary fragments. And the idea is that we then can generate these sort of unique and new natural product fragment combinations. And the key here is that the sort of the affinity towards steroid transport proteins should come from these primary fragments. And then the sort of selectivity should come then from the appendage of these diverse secondary fragments. So this, we were hoping that by synthesizing these different fragment combinations, we would be able to access both potent but also selective inhibitors of steroid transporters. And this kind of approach couples, you could call it pseudo-natural product, BIOS, and also some elements of DOS. So we have really sort of merged strategies here in this approach. We have some criteria when we make these compounds. So we try to keep the step count low. We try to focus on scaffold fusions that are sort of unprecedented. We always try and make three to five analogs per combination, simply because we found that this is sort of necessary, but also sufficient to find a starting point, basically, that we can then do medicinal chemistry on for any given scaffold combination. We do this racemically. We'll get to why that is in a second, but it's essentially in short, we can sort of double our compound library if we have both enantiomers present. And we, of course, want to do this with common building blocks, again, to keep the step count down and to make the synthesis sort of accessible. So I'm going to jump straight into a couple of examples from the lab, and this one here is published, so I'll go through it quite quickly. But we started by synthesizing this key kind of hydroxy ketone. This is done in 6 steps with just two column chromatographies, and this is a very highly optimized procedure now. But starting from this ketone, we can basically use this as a nice functional handle to access sort of a wide variety of different heterocyclic scaffold fusions. Of course, we can do Friedlander quinoline syntheses, we can do Fischer-Indol, we can do sort of aza-indol syntheses, ring expansions, and also sort of spirocycle formation here. And this is quite nice because this is a very versatile building block that can access the sort of diverse scaffold fusions via different heterocyclic chemistry. We're not limited, of course, to the ketone. We can also, from alpha bromoketones, make 3 other heterocycles, thiazoles, and quinoxylines, for example, here as well. And these can also be accessed in moderate to good yields. And these procedures are very classical, and they work very well on these scaffolds. We also access in a similar way from the ketone the hydroxymethylene shown here, and either protected or unprotected. And here we can react this both with aryl hydrazines to give a mixture of regioisomers here of these pyrazoles. And while normally it's not maybe not ideal to access the, sort of a mixture of the two, again this is quite useful from a compound library point of view. And as you'll see in a couple of slides, that actually the position of this aryl substituents actually quite significantly impacts the biological activity of these compounds. So again, we can access different compounds in just one synthetic step here. And the same also goes for these other heterocycle synthesis from these sort of pyrazolo pyrimidines over here. We can access both different regioisomers in the so-called angular one and also the linear one. Finally, we can make unsubstituted pyrazoles and also ones from the hydroxylamine over here as well. Okay. So this is published, so I'm not going to spend too much time on this, but we wanted to go beyond what we called edge-fused type of scaffolds, and we did this in a follow-up story which is not published, so I'm going to spend a little bit more time on this. And here, one of the sort of guiding principles is that also we can, to change basically the stereochemistry of this primary building block. So I showed in the first part that we had this sort of transdecalin system, which sort of more closely mimics cholesterol itself. But actually, if you take cholic acid, we have this cis-fused decalin system, which gives a quite a stark sort of difference in three-dimensional structure of these compounds. So we can make the same kind of edge-fused or spirocyclic fused compounds as we did previously using the scaffold fusion. But here we also wanted to apply a sort of a second layer of diversity and complexity by sort of doing ring distortion and modification strategies on these, particularly on this on this indole scaffold here. And so we did this. So starting from these, so a variety of substituted indoles, we can actually do basically simply by modifying the oxidative sort of conditions, we can do both oxidative ring expansion here. So you can open up this indole to give you a ketolactam here. This is quite a nice way to make these medium rings. And these can then be recyclized to give a sort of a ring contracted quinolone here, which is also quite a nice kind of a new scaffold fusion that we hadn't targeted before. Then by using, in this case, oxone rather than periodate, you can simply make these hydroxyindolenines, such as here, where we don't have a sort of a ring opening. We can also do a ring contraction to give you this this spirocyclic oxindole here, and again this is slightly also with oxone, but by simply by changing the solvent. So going from water and acetonitrile to basically adding acetic acid and THF, we can actually get a different sort of oxidation product. And finally, we have this very interesting scaffold down here. And I'll come to what's going on there in a second, but it was a little bit of a surprise to us. But just to make sure you all believe me, we have crystal structures of all of these to prove that this is exactly what is going on. So with this compound, this kind of quite unusual looking compound on here, so what we were really intending to do, so this was a sort of serendipitous discovery, because what we really wanted to do was as an oxidative ring contraction to give the spiroxindole, and this would work first with NBS, where you can sort of brominate the indole, followed by basically a semi-pinnacle rearrangement, which would give us the basically the desired or the originally intended spiroxindole, which is the one shown here. However, so we're still an acid here, so this can be protonated. But if you actually redraw this in 3D, you can see that now, because of this cis-fused sort of decalin ring system, it's no longer a decalin, but it was originally. You can see that now this hydroxy group is perfectly positioned to do basically a condensation reaction here, and that is exactly what happens. So you basically lose water to get this spiroxypino indole, which is sort of shown over here. So this was an unintended reaction, but the reaction actually works very well, and it works with a range of different substitutions on the indole. So quite a range of analogs was accessed using this approach. Compared to the rest of the library, this is also a lot more sort of three-dimensional in structure and shape, which we were interested to see what the biological activity of this would be. So to cut a long story short, we also this is the kind of the library to date. So we have these 3 different boxes. So the one on the top left here is from the transdecalin scaffold. This is now published. We also have some from an estrogen derived sort of primary scaffold here, which is also published. And here we have from the cholic acid cis-fused decalin system. This is about to be uploaded to ChemArchive in the next couple of weeks. So this will be out very soon as well. So our total library is we have over 500 sort of sterile inspired compounds. So and we have been able to screen these quite broadly, and I'll get to that in a second. So as I mentioned right at the beginning of the talk, we are interested in finding compounds that inhibit sterile transporters. So a lot of work in my lab is also done on protein biochemistry. So we have basically expressed and purified all basically mammalian cholesterol transport proteins, and we developed 2 assays to test our compounds. We both have a differential scanning fluorimetry, or DSF assay, also known as a thermal shift assay, where we can see basically the melting curve of our cholesterol transport protein, and this can be stabilized where we see a thermal shift in presence of an inhibitor. So this is a nice binding assay which tells us if our compounds bind to the sterile transporter. We also have a more functional assay where we have a fluorescently tagged cholesterol here, 22-NBD cholesterol, which when bound to a sterile transport protein will rotate more slowly. And when you shine plain polarized light, you will have high retention of polarization. But when you then inhibit with an inhibitor shown in green here, this will be displaced. You have more rapid rotation and more depolarization and low FP signal. So we can basically determine whether a compound is able to inhibit the interaction of fluorescently tagged cholesterol to a cholesterol transporter. And so one family that we focused on, or that I will focus on today, is the Aster family of cholesterol transporters. And these are important in transporting cholesterol from the plasma membrane to the ER. And it's also been shown that they are important for a cellular process called autophagy, which is a cellular recycling process that is actually quite important in cancer cell survival. So we had an interest from a biological point of view of developing inhibitors of particularly Aster A. There is a known inhibitor already available, but there's always need for more compounds, and we also wanted to identify inhibitors of Aster B and C, which at the time were also not known. So the first thing we did is we used our thermal shift assay to test this was the first generation library. So as I said, we have 500 compounds now, but at the time we had, this was only 65. But we found several active compounds which sort of passed our activity threshold. And I'll focus on one here, which we called AsterSyn1. So this is an inhibitor of the protein Aster C. And we can see here that there's a very clear shift in the melting curve from the DMSO control in green to the compound in pink of about 2 degrees. We also repeated the screen here for Aster A. So the top was Aster C and the bottom is Aster A. And here we found a new compound also, which we called asterpirin, which had a very nice stabilization of 5 degrees of Aster A. And immediately what's interesting is that you can see that these both have the same primary steroidal scaffold, but have a different secondary scaffold here with the angular pyrazole compared to this linear pyrazolopyrimidine. And that gives quite a difference in sort of activity and selectivity between these cholesterol transporters. And we looked at the selectivity starting off from AsterSyn1, and as I mentioned earlier, we made this compound racemically initially, and here we can see that it has a nice selectivity about about 20 to 30 fold over Aster A and B. So the pink is for Aster C. But then what we did is we actually synthesized by an asymmetric Robinson annulation, basically the building block in enantioselective fashion. And we could actually show that surprisingly, the one with the unnatural stereochemistry compared to sort of cholesterol shown up here was actually both the more potent, but also the more selective compound. So this kind of validates the approach of making the compound library in a racemic fashion, because actually had we chosen the compound with the natural stereochemistry, this would have been both less potent, but also much less selective for Aster C over Aster A, and we might not have continued with this approach. So in the end, what we have is a very potent Aster C inhibitor, which is very, very selective over B and A. What we've also looked at as a sort of more functional assay is a transport assay where we use liposomes, where we have basically fluorescent lipids, one with cholesterol and one with a second lipid, which are a FRET pair. And so basically, when you add an acceptor liposome, which is non-fluorescent, your protein can extract the fluorescent cholesterol from the donor liposome and transfer it to the acceptor liposome. But this process can be inhibited with our inhibitors. So we expect to have a slower loss of Fred signal when this process is inhibited. So we did some medicinal chemistry optimization from astrocin 1, and we could show that a couple of positions were poorly tolerated, and actually substitution around this aryl ring was generally sort of well tolerated. And with particularly the addition of a chloro here gave a more potent compound with increased selectivity. So this was, it's not a big change, but it was still sort of significantly useful that we continued work with astrocin 2. And here we can see in our sterile transport FRET assay, we can see that for Aster A and Aster B, even in presence of the astrocin 2, we basically rapidly have a loss of this FRET signal in both cases, which is in line with the vehicle control. So there's no inhibition here of sterile transport. But in Aster C, if we take astrocin 2 at 1 micromolar, we almost have complete inhibition of the sterile transport effect. So this also confirms that this is a very nice, potent and selective inhibitor of the sterile transporter Aster C. If we come back to this asterpirin that we also discovered, this was also synthesized as a racemic mixture, but then we synthesized both enantiomers. And again, we could find that so in this case, it was interesting because the natural steroidal stereochemistry was also the one that was more potent. So this is what I would have expected in general. And this compound is very selective for Aster A over B, a little bit less over C, but if you look at the sterile transport assay data, we can see nice inhibition of A and almost no effect on B and C. So at least in this more functional assay, this selectivity looks to be pretty good, useful for a sort of further biological experiments. So I wanted to come back before we finish up, just to this interesting compound over here which we called asteroxin. So this was the unexpected spirox indole, which condensed with the hydroxyl group on the A ring of the sterile system, and we had, as I mentioned, we were able to synthesize quite a lot of analogs also from the start and also beyond with substituents on this aryl ring, and to our great surprise, basically, because this compound doesn't really look steroidal anymore. In fact, it's very structurally very different. We actually identified a compound, which this one here, asteroxin, that was very potent and also completely selective for Aster A over B and C. So if we look at basically these other compounds in the series, they are all, so this asteroxin 1 was the most potent, but the selectivity was generally maintained also through the other analogs. One of them here, 1D, picks up a little bit of activity against another sterile transporter, but in general, the selectivity is really excellent, especially for asteroxin 1. And so this was really a great surprise, but very interesting for us. And again, this library was also made as racemic mixtures. So we sort of predicted that the one made from the unnatural stereochemistry. So in this case, the SRSS asteroxin 1 would be the most active. And this is exactly what we saw. So basically, this is the minus asteroxin in red is the most potent one. It's more potent than the racemic mixture in sort of purple. Just that it's about twofold increase. And then the other enantiomer is much less potent. So it's almost 2 orders of magnitude less active, which also is in line with some modeling that we did. So this is not a crystal structure, but there is a crystal structure of Aster A, and actually we can see that this both the potency and the sort of selectivity comes with. We only see it with this particular enantiomer. And although cholesterol is a fairly flat molecule, actually the binding site, especially for Aster A, is not that flat. So it can accommodate this slightly more 3-dimensional structure, which actually fills out the pocket in a more efficient way than maybe even cholesterol itself. And so that we think it explains basically the potency of this compound. And the selectivity in part comes from this, but also we believe from a series of hydrogen bonds here at the sort of towards the exit of the pocket that we specifically see with the morpholine, and actually not with some of the other analogs. So that's kind of where our group's research is for now. And I just wanted to say, if anyone is interested in this kind of approach, if you're, for example, if you're a chemist and you're working on sterols, if you're interested in screening any of your compounds in a panel of sterile transporters, as you can see, we have a lot of different assays for pretty much all except one of the human sterile transporters. So please feel free to reach out if you want any of your compounds screened in the panel, and we're always very excited to collaborate. And we have done this with multiple groups now, and we get some really interesting results that we are following up on. So please drop me an email if you're interested in this. So just to wrap up, I'd like to thank my group for being a really fantastic group of researchers. In particular, the work that I talked about today was done by Thomas, who I believe is also online listening in. So thanks, Thomas, for especially for the first part of the sterile library. Frederick did the spiroxypino indole, so the second cholic acid inspired library, and Laura did a lot of the biophysics and screening also in the group. And then finally, also thanks to all the funding bodies who support our research. And thank you for joining in today. And I'm happy to take any questions that you may have. So thanks.
Thank you, Dr. Luca, for your interesting talk and sterile chemistry, basically. So there is one question. In fact, there are two questions from Pratibha. How you select the racemic mixtures of analog to determine its efficacy for biological activity? And second part to it is this phytochemically fits with the targets?
Yes. So for the first part, basically, so as I mentioned in the talk, we make all the libraries are always racemic, both to double the library size, but also because actually, even though we might expect that the active compound would come with ones that have similar stereochemistry to cholesterol itself, as it turns out, this was actually not the case. And we have multiple examples now where sort of inverting the stereochemistry is actually beneficial. We actually have so in terms of the how we can rationalize that with the binding site. So for the aster C ligand, which was the one that actually has the sort of the opposite stereochemistry to cholesterol, we actually our modeling suggests that that actually it doesn't fit in the binding site in the sort of expected way, where we have the sort of the three hydroxy group sitting at the bottom of the pocket, but it actually sits in a slightly flipped orientation. So this is something that we couldn't necessarily have predicted in advance, but that also kind of strengthens validates basically the approach of doing everything racemically. But we are also in the process of getting trying to get crystal structures for some of these, so that we can confirm the binding modes beyond just doing molecular modeling. I hope that answers the question.
Dr. Bhatia, you said there are another question. What is the second question? I can read out the second questions here. Biosynthetic diverse secondary fragment part. How is it relevant in designing new cholesterol transporter in the future?
Yeah, it's a good question. So basically, the way the library is designed is that we have the sort of affinity towards sterile transporters, of course, comes from the primary fragment. So this is the part that looks like a sterile, basically. But one big challenge that we have is, of course, all of these proteins bind cholesterol, right? So they have very similar affinities for cholesterol. So if we take compounds that are sort of too similar to cholesterol, then we may run into selectivity issues. So it's the secondary fragment that should basically give us selectivity for one sterile transport protein over the other. And actually, I've shown two examples of this with the astersin and also the asterpirin, where simply by changing the secondary fragment from, for example, the pyrazole to the pyrazolopyrimidine, we sort of completely changed the selectivity of these compounds. So we have more unpublished examples of this where we see this again and again, that basically simply by changing, so we keep the primary fragment identical and we just change the secondary fragment and we get a completely different selectivity profile. So that even beyond maybe what I would have expected when we started this project, it was kind of nice for us to see this, because of course that was the idea, but sometimes these ideas don't necessarily work out in practice. But we really see this time and time again, that simply by changing a secondary fragment, we completely change the selectivity across the sterile transporters. And that also comes from the fact that these transporters, they are similar, but of course they are not identical. So there are certain residues within the binding site where you can pick up additional interactions, whether these be hydrogen bonding or slightly different sort of shape complementarity in the binding site, which enables us to do this.
All right, I think there are 2 questions from the chat box. I have a quick question, Dr. Laraia.
Yes.
Hello.
Yeah. Can you hear me?
So, I always wonder, like, for example, if you got a natural product as your very potent molecule, and that could be a really potential drug. Let's say when it is required in very large quantities, and if the natural sources is very limited, do you think is it very easy or challenging to prepare the compounds in kilogram level, maybe pilot plants, several kilogram levels? So do you think some sorts of challenge is involved there in terms of process? Yep.
Yeah. So I guess you're referring to the compounds that we have made, or to natural products themselves.
In general,
Okay.
I'm asking. Yep.
Yeah, so I think for the ones that we have made, I think so. Now the procedure is very streamlined. So as I said, we can do the first 6 steps with currently with just 2 columns. But I think actually we're working to get this down to just 1 column at the end. So actually we can bring through. So the key building block, the key sort of ketone building block, we can now almost make that with just with a couple of workups basically, and with only 1 purification at the end. And then from that you can basically access all of these diverse structures. So that I think is very amenable to scale up, and we do most of those steps on let's say 20 gram scale or something. So of course I'm not sure how we would scale to ton scale, but we can certainly do it on a fairly large scale. But yeah, I think this is one of the main issues that especially with these sterile transporters, is that there are a few natural product ligands that are very potent inhibitors, but as I mentioned, these are 50-step total synthesis. So kind of the goal behind our research is really to move we want to keep this natural product element that will give us the sort of high binding affinity and so on, but we want to move to structures that are synthetically more accessible. And I think all of the compounds that we've made are under 10 synthetic steps and with very few purifications. So they are more amenable to kind of large scale synthesis and production. We are not there yet, but the potential is there, I would say.
All right, great. Thank you very much, Dr. Laraia, for the nice next talk.
Thank you.
Okay, so now we'll move to the next speaker, Dr. Neelangela Majumdar from CDRI, Central Drug Research Institute in India. It's my pleasure to introduce her to this audience. She had a BSc degree from an university, Vishwabharati. It's very good to see that I also had a master's degree from the same university. After bachelor's degree, she had a master's from IIT Kharagpur. Then she moved to USA for PhD in chemistry, working in Michigan State University with Professor William D. Wolf. After finishing PhD in 2012, she moved to Japan, Tokyo, for first postdoctoral research experience with Professor Shibasaki's group, JSPS Fellowship. Subsequently, she had another postdoctoral experience with Professor Benjamin List, a Nobel laureate, in Max Planck. Finally, she returned back to India and joined CDRI in the year 2018. Currently, she is working as a senior scientist in medicinal and process chemistry division. With this short introduction, I request Dr. Majumdar to present her talk.
Thank you, Professor Laha, for your kind introduction. Am I audible and my slides are visible? Please confirm.
Yep.
Okay.
Much, yep.
Thank you so much. Good morning and good afternoon, everyone. Before I start, I would like to thank Dr. Rohit Bhatia as well as Thieme for inviting me to give this talk on this great platform and hosting this very nice Cheminar. It is very informative and I think it will be very useful. So today the title of my talk is Unactivated Carboxylic Acids in Catalytic Asymmetric Ring Opening Reactions. From the title, just a moment. So from the title, you can understand that this work is about simple carboxylic acids and their role in some asymmetric ring opening reactions. My slide is not moving. Just 1 second. Okay. So the general scheme is shown here, where you can see the carboxylic acid acts as a nucleophile in this catalytic asymmetric ring opening transformation. When we first started this chemistry, we actually screened a lot of metal catalysts in combination with a wide variety of chiral ligands. But after thorough screening, we found that iridium catalyst actually works best in combination with carrier ligand for this particular reactions. Generally, very low catalyst loading is required to achieve high yield and excellent enantioselectivity for this ring open products that can be isolated as single regioisomers. Now the question is, what is new about this reaction? First of all, I would like to draw your attention to this double bond that is attached to this three-membered ring. Because of this double bond, in presence of transition metal catalyst, this ring opens up following the formation of this metal pi-allyl complex, and this results in this dipolar zwitterionic species that can then participate in a range of various cycloaddition reactions. And this is a very well established chemistry. A lot of asymmetric variants are also available in the literature. On the other hand, simple ring opening reactions, nucleophilic ring opening reactions, are not so common for these substrates, especially the asymmetric reactions. Secondly, if we consider the nucleophilicity, carbon nucleophiles are definitely on the top of the list compared to nitrogen or oxygen nucleophiles. In fact, simple free unactivated carboxylic acids are considered as one of the weakest nucleophiles. So here we have shown such free carboxylic acids acting as efficient nucleophiles in this catalytic asymmetric ring opening reaction. One important challenge for this reaction is the regioselectivity. As you can see in this nucleophilic ring opening transformation, 3 regioisomeric products are possible that can form via nucleophilic attack via path A, B, or C. In this case, these 2 products are particularly possible because this reaction occurs via this metal pi-allyl complex formation, either by inner sphere mechanism or by outer sphere mechanism. In case of inner sphere mechanism, then after the pi-allyl complex formation, the nucleophile coordinates with the metal center, and then the reductive elimination takes place from the carbon. That base stabilizes the positive charge, leading to the formation of this branch product. On the other hand, in case of outer sphere mechanism, there is no pre-coordination between the metal and the nucleophile, and the nucleophile prefers to attack at the least hindered carbon, forming the linear product. Although it is not a specific rule, but generally iridium catalysis favors the inner sphere mechanism, and thus the branch product formation, and palladium catalysis favors the outer sphere mechanism, forming the linear product. Since in this reaction we are using the iridium catalyst, this actually solves our regioselectivity problem because mostly we observe the branch product formation. So today I'll be talking about the catalytic asymmetric ring opening of vinyl oxidants, vinyl cyclopropanes, and vinyl aziridines. Excuse me. Let me first start with vinyl oxidants. These substrates are either commercially available or quite easy to prepare. On the other hand, carboxylic acids are inexpensive class of compounds that are stable and commonly available. They are also considered as feedstock chemicals. This reaction requires very low catalyst loading. Generally 0.25 mole% catalyst is sufficient in most of the cases. We have varied a large number of examples, and these chiral vinylic diol compounds can be obtained in up to quantitative yield and greater than 99% enantioselectivity. So here some substrate scope has been shown. You can see that we shown the variation of aromatic as well as aliphatic carboxylic acids. We have also varied differently substituted vinyl oxidants, and throughout the substrate scope, yield and EE both are quite impressive. We can access both the enantiomers of the product by simply changing the configuration of the ligand. And it is important to note here, in the absence of this vinylic double bond in the oxidant substrate, the reaction just doesn't work. This method has a very good application potential. The reaction can be performed in gram scale, and this enantiopure product can be obtained in quantitative yield. Now, this chiral monoester derivative can be easily converted to this chiral vinylic diol and then to chiral vinyl oxidant without any loss of EE. These two compounds are actually very important asymmetric building blocks that are sometimes this chiral vinyl oxidant or chiral vinyl diol. You can see in many they are used as a starting material for many bioactive natural products or medicinally important compounds. But these two compounds are with very limited commercial availability. So this vinyl diol actually can be used in a very short and efficient synthesis of goitrine and epigoitrine. These two are medicinally very important natural products, and they are enantiomeric to each other. Again, this vinylic chiral diol compound can be easily converted to various ring-sized chiral lactones in simple synthetic steps, and these chiral lactones can be part of many important biologically important natural products. We have also studied the possibility of kinetic resolution in this reaction, and we found that with time and conversion, enantioselectivity is constantly increasing in the recovered vinyl oxidant substrate. So these results indicate that kinetic resolution is definitely involved in this reaction. Now I will move on to the next topic, which is the catalytic asymmetric ring opening of vinyl cyclopropanes. As I have already discussed earlier, that the cycloaddition chemistry is very, very common for this kind of substrates, but simple nucleophilic ring opening is not so common, especially asymmetric examples. They are not so much available. In fact, this work is the first report for the catalytic asymmetric ring opening of vinyl cyclopropanes using oxygen nucleophile. Here we have used a free carboxylic acid as the oxygen nucleophile, and a wide variation of substrates have been tried to achieve high yield and very high enantioselectivity. The selected examples of the substrates have been shown here. We faced many challenges while developing this chemistry, and one major challenge that we faced was to determine the exact stereo configuration of this catalytic reaction product, because this kind of chiral compounds were not reported in the literature, so we could not compare the optical rotation with any known molecule. And also these kind of compounds are very oily in nature, so it was quite challenging for us to get the crystal structure for these products. So we started exploring a lot of reactions for the derivatization of this chiral compounds, so that we can get a nice crystalline solid. During one of those experiments, it's a very nice story, that's why I'm sharing this. During one such experiment, where we wanted to do metathesis reaction between one of this catalytic product and vinyl quinoline in presence of Grubbs catalyst, we found that the reaction worked perfectly, giving a single product formation. So initially we thought that this must be our metathesis product, but after complete analysis, we found that it's actually not the metathesis compound, but it's a hydroalkylation product. This result was quite surprising to us, and it indicated unconventional reactivity of Grubbs catalyst. So we developed this chemistry separately, where hydroalkylation overrides metathesis. Now coming back to the determination of stereo configuration, after a lot of experiments and thorough investigation, we found that one of this catalytic reaction product can be conveniently converted or cyclized to the corresponding chiral vinyl cyclopropane without any loss of EE. And this chiral vinyl cyclopropane is a very nice crystalline solid, so we could get our desired crystal structure and determine the stereo configuration of the catalytic products. Now I will move on to the last part of my talk, which is the catalytic asymmetric ring opening of vinyl aziridines. This work is almost complete, and we are going to communicate it very soon. We have filed a patent on this work, and this reaction is actually a great example of sustainable catalysis. So for the sustainability in chemistry, we generally look for several factors. For example, cost efficiency, atom economy in the process. We also try to avoid toxic or hazardous chemicals. And for catalytic reactions, turnover number is a very important factor. For this enantioselective ring opening of vinyl aziridines using free carboxylic acids as the nucleophiles, we could reduce the catalyst loading to ppm level to achieve very high yield, excellent regio and enantioselectivity in a large number of examples. So less catalyst means basically use of less use of precious iridium metal. It also indicates use of less use of precious chiral ligand and less metal contamination in the later stages of the synthesis. You may also notice that this product is actually a chiral beta-amino alcohol, which is a very important motif found in many bioactive natural products and medicinally important compounds. The added advantage of this product is this vinylic double bond that can be used for further synthetic transformations. So here some selected examples are shown. We mostly developed this strategy or the substrate scope using tosyl protected vinyl aziridine, but we thought that tosyl group can be sometimes very hard to deprotect. It can be very problematic. So we thought of using some other protecting groups, and BOC group actually worked pretty well. This particular substrate has been developed quite recently, and we found that this substrate worked quite nicely, giving almost 100% enantioselectivity single enantiomer with high yield. And here you can see that we can access both the enantiomers of the product by changing S ligand or R ligand, and the reaction works very well in gram scale also. So we varied a lot of different carboxylic acids in this reaction, aromatic as well as aliphatic carboxylic acids. We have also varied the substitutions on the vinyl aziridine, and we found that even for a sterically hindered aziridine substrate, the reaction works pretty well, and the stereocenter can be formed quite efficiently with high yield and excellent enantioselectivity. For the synthetic applications, we have shown, I think, the shortest synthesis of goitrin and epigoitrin almost in 2 steps, starting with this catalytic reaction product. So the first step is the tosyl deprotection that I was talking about. Actually, it was pretty challenging for us initially because a lot of conventional reaction conditions were not working. So after some exploration, we found that samarium iodide works pretty well for this reaction. And then by deprotecting the acyl groups and then reacting with TCDI forms the goitrine or epigoitrine in high yield. We could also cyclize this catalytic product to the corresponding chiral vinyl aziridine. But in this process, some of the enantioselectivity got compromised because we started from almost pure enantiomer and we got 92% E in the product. Before talking about the other important synthetic application of this method, I would like to refresh your memory about Garner aldehyde. This has been used as a versatile chiral intermediate it has been used for a long time in a lot of important total synthesis of natural products as well as medicinally important compounds. This is the structure, and here nitrogen for this amino alcohol, nitrogen is directly attached to the chiral center. Now we thought about the complementary structure. What about this complementary structure? This is also an amino alcohol, but here oxygen is directly attached to the chiral center. This was never reported before. So we thought of approaching this synthesis, and we started with this catalytic reaction product. First by acyl deprotection and then by acetal protection of this amino alcohol. This double bond was then oxidized to the carboxylic acid and then converted to the Weinreb-amide. Then a simple diol H reduction forms this aldehyde. This aldehyde was not isolable in the pure form. So we used the crude mixture in the next olefination step and could get this olefin product successfully. This is so finally we could achieve this the synthesis of this aldehyde, but this is a little bit of a lengthy process. So we thought of we think that ozonolysis can solve this problem from this acetal protected amino alcohol. But currently we don't have the ozonolysis facility with us, so we could not do this reaction, but I think it will minimize the steps quite significantly. So to summarize, I would like to say that I have shown you today the catalytic asymmetric ring opening of vinyl oxidants, vinyl cyclopropanes and vinyl aziridines using iridium catalysis and using free unactivated carboxylic acids in all these cases as very efficient nucleophiles, and the products are obtained in high yield and excellent regio and enantioselectivity. It should be noted that these products are actually of high value because they can be used as a very important synthetic intermediates for making complex bioactive molecules. Finally, I would like to thank my students who really worked hard and sincerely for developing this chemistry, and the funding agency as well as my institute for all the support. And finally, thank you all for your kind attention.
Thank you,
Rohit.
Dr. Nilan.
Yep.
Yes, please, Professor Laha.
Yeah, go ahead.
Thank you, Dr. Nilanjana, for your nice talk. So there are two questions in chat box. First one, is there any example where the palladium can perform oxidant ring opening through Sujit-Trott kind of reaction?
Sujit-Trott, yeah.
Yeah. And the second question is, can this methodology be applied for epoxide analogues of internal olefins?
First of all, for the first question, yes, of course, palladium catalyst has been used, but as I have mentioned that generally for palladium catalysis, it will go through the outer sphere mechanism, and in such case, only linear product will form. And in case of linear products, probably we will lose the stereocenter. So that is the answer for the first question. And now for the internal olefins of the epoxide, for the epoxide analogs, of course, yeah, they can be used, but regioselectivity could be a problem in such case.
Dr. Rohit, do you have no other question?
Yes.
Yeah, there's another one. Yep. Go ahead.
Yes. Then there is a question, can these be used as antimicrobials?
So since we are in Drug Research Institute, we are actually going to screen these compounds for different disease areas, of course, but it has not been done yet. We intend to do that. Then it will be more clear, I guess.
Then there is a question, ozonolysis might contribute to undesired oxidation and ring opening. A standard Lyman-Johnson oxidation may be helpful. Mild oxidation of olefins to aldehydes. I think
yeah, so that's a suggestion. Maybe we can try it.
Yes, this is a suggestion, not the question.
Yeah.
Yeah, then there is one more question. Have you tried all the substituted cyclopropanes for ring opening?
All the substituted cyclopropanes for ring opening. Actually, I showed only how to say, I showed only limited examples or selected examples. We have shown okay, substituted cyclopropanes. No, we did not try all the substituted cyclopropanes. It should be only vinyl substituted cyclopropane. Otherwise, the reaction might not work. Vinyl group is very important for this reaction, for making the pi-allyl complex.
Okay. So there is one more question. What is the cost of catalyst and is it useful for a commercial scale?
Yeah, iridium is a precious metal, and of course it is expensive catalyst, but as I have shown in our reactions, we could reduce the catalyst loading significantly, for example 0.25 mole% to ppm level catalysis. So I think in such case definitely it can be useful for commercial scale if the catalyst loading can be reduced like that.
Thank you, Anand. The next question is to check the stereochemical outcome of chiral alcohols. You can try to prepare it as a mandolate followed by NMR. Okay, this is suggestion. Sorry.
Yeah, I think we tried actually, but it was not really confirmatory. Yeah.
Okay. What is the advantage of azidrine ring opening product as against the diol product for medicinal chemistry? So this is the last question we are taking.
Yeah. So a diol product is completely different and amino alcohol is completely different. What we can achieve through amino alcohol, of course, we cannot achieve through diol. For azidrine chemistry, the important part of amino alcohol, let me show my slide, go back to my slide again. So if you can see on this slide, this was a supportive slide actually. And you can see that this kind of amino alcohol where nitrogen is directly attached to the chiral center, that can be easily obtained from naturally occurring chiral amino acids via chiral pool synthesis. But what we are doing here actually, but if we want to attach oxygen directly to the chiral center, then it is quite challenging, because these kind of reactions are not reported in enantioselective manner, and they don't give very high enantioselectivity as well. But in our reaction,
Professor.
hello. So also for achieving this kind of vinyl substituted products, we have to start from acrolein. Acrolein is quite unstable, toxic, and with limited commercial availability. So in such case, another thing is TMS cyanide is also very, very toxic. So in such case, I think vinyl azidrine ring opening would be a very good approach, complementary approach to get this kind of chiral products that are otherwise difficult to obtain in a straightforward manner. I hope that clarifies the question a little bit.
Thank you for your reply, Professor Dr. Naranjana. Now we start with the Professor Laha. We can start with the next talk.
Yeah, Dr. Naranjana, just I have a quick question, if you don't mind.
Yeah, sure.
In the first part of your presentation, you talked about the use of the epoxide and the ring opening of epoxide.
Yes.
And you also showed a compound that is available in Aldrich. So what was the scale? What was the scale for this reaction?
This
Yes.
was yeah. So this we have done only up to is the slide visible?
Yes, yeah, let's see, that's
Yeah,
1.4.
so we have made up to this compound. This is actually a well-known process reported in this paper, so we did not do this reaction, but this up to this part we have done up to 1 gram scale. And for millimole substrate scope, like for the other substrate scope in the reaction, we have used 0.25 mole% catalyst, but when we did the reaction in 1 gram scale, we could use only 0.2 mole% catalyst. So I think that if we scale up the reaction, catalyst loading can be reduced much further.
Okay. Does it require any globe box, use of globe
No,
box,
no,
or?
no. That's a very good advantage of this reaction. It doesn't require any globe box. In fact, we don't even need to do any column purification of this product. We just do simple filtration, and we can get
Great.
completely pure product.
Yeah, great. Thank you very much for
Thank
an
you.
excellent talk. Very good insight. Now we'll move to the third speaker of this event, Dr. G. Sudhakar. It's my pleasure to introduce him to this audience. As a master's degree from Osmania University, Hyderabad, and then PhD from IICT Hyderabad, and then he has a one brief postdoc in US in Purdue University with Professor Arun K. Ghosh, and then he returned back to India, took up a scientist position at IICT Hyderabad, and he has been promoted to principal scientist and currently working as principal scientist. Please, Dr. Sudhakar.
Yeah, thank you, Professor Laha. My slides are visible. Am I audible, please?
Yes, both.
Okay. Yeah, thank you very much. Good morning, good afternoon. First of all, I would like to thank this organizing committee, Thieme team, for giving this wonderful opportunity to present in this webinar, Cheminar. So my title of the topic is, I'm just going to show that, little different how we got this product. So the role of synthetic steps order in developing key intermediates or key starting materials, active pharmaceutical ingredients. So I'll start with a few areas, actually the research overview, and that a very brief about some electrocyclation reactions, and some 1 or 2 slides about this totasis of macro and indole alkaloids, and today topic more on the synthesis of ladipasvir and efflogens. So coming to our research overview, if you see, we work in various research areas. One is electrocyclation reactions and natural product synthesis, and whatever the new chemical entities we obtain, we evaluate for the medicinal chemistry aspects, and the protocols we developed in this organic synthesis and the experience we gain. So we use it for a process for APIs. And this all research projects are supported by various funding agencies that include Science and Engineering Research Board and CFIPRA and our organization, CSIR, IACT, and even Ministry of Earth Sciences, and also we had some projects from the industries. So coming to the electrocyclation precursors, more specifically natricyclation, in which divinyl ketone in the presence of acid gives pentadienyl cation, which undergoes 4,5 conrotatory electrocyclation to provide this auxiliary cation, and then loss of proton provides cyclopentanone. So irrespective of the starting material, once the pentadienyl cation is generated, this is actually requisite for this conrotatory electrocyclation. So we consider if we can generate this pentadienyl cation from novel electrocyclation precursors, so that could be complementary for this existing natricyclation. So we have developed several this electrocyclation precursors, starting from this divinyl oxirane, divinyl aziridine, and even divinyl donor acceptor cyclopropane. Just give a minute to show the pointer. Yeah. So even aryl vinyl cross conjugated triene, and recently we have used even aryl vinyl this oxetane as well. So they have provided a variety of compounds, including this indonyl oxypines and some indonyl compounds, and highly substituted indonyl compounds, and including some indonyl glycines and some tricyclic, bicyclic compounds, including some carapinol, the natural product derivatives. So these are some representative publications from this area. Coming to natural product, we also actively participated for several years in synthesis of natural products. So our aim is actually synthesis of this natural product by developing a novel synthetic strategies to access group of or a class of molecules, and very
Dr. Sudhakar, you are not audible.
Let's just wait a minute for Dr. Sudhakar's connection to resume.
Okay, all right.
Looks like Dr. Sudhakar's connection has dropped. I'm sure he'll be back in just a second. People want to add their questions to the chat. We can use these couple of minutes productively. Apologies for the interruption of service, everyone.
Oh, no problem.
Badwa, if we can't get Dr. Sudhakar back, we may need to move on to the next talk and resume Dr. Sudhakar afterwards.
Yeah, we can move to Dr. Swain, and then Dr. Sudhakar can join us again.
Okay, all right. So then I invite, So I invite Dr. Swain from NIPER, Kolkata, who is currently an associate professor there. He has several years of working experience in industry. He worked on various, organic synthesis, drug discovery, drug design, and on all. So, PhD from National Taishan Hua University and a postdoc from University of Texas. So currently he's an associate professor in NIPER, Kolkata. I invite Dr. Swain to give his talk. Thank you.
We can't hear you, Dr. Swain.
You're audible, no?
Yeah.
Yep.
Now,
Is the slide visible?
yes.
Yes.
Okay, fine. Good afternoon, all, and thank you, Thieme team and Professor Laha for the invitation. And today my talk will be on photocatalyzed continuous flow synthesis of pharmaceuticals. And so first I go to the basics, little background about the continuous flow synthesis, because many of the audience they may not have the deep knowledge how it works. So some slides will be in the background. Then we move to the real agenda. So this is the basics of this diagram of this how it works. The system works. We'll have the reaction rather than a traditional flask on the reactor. We'll have the tubular reactors, most tubular or cartridge type reactors, and this is the reactor system, is a schematic diagram of a reactor, and you can pump through the microtubes the reagents or the reagent plus in the solvent system from two pumps, and you can have multiple pumps also. A single pump depends on your requirements. So the reactors, it happens, you can do room temperature, it can happen in cooling as well as in the heating condition, and also in the heating beyond the boiling point of the solvent. And we can use back pressure regulators to do the pressure reaction in the system and get the compounds out and collected. And this is how the flow reactors have advantages over the traditional process. This is the recent process research development 2020. Marcus Bowman, it is from Ireland. The team he has published on recent review articles. So I take the picture. So it is faster compared to the traditional process. So it is safe, and also some challenging reactions can be easily operated in this. And the quality of the product, we talked about the impurity profile of the product, and also smaller footprint. So we can try from small milligram scale up to the scalable also. It depends on our requirement. We can change the batch size. So this is more detailed description of the what kind of reactions we talked about. The first is safety parameters. If we talked about, we'll see this corrosive reactions like fluorination, nitration, and the cyanide reactions. We can do it very efficiently. And pressure reaction, like which is very pyrophoric, the hydrogenation reaction we can do easily with pressure. And also it is very safe for the high temperature exothermic reactions. Including we can do new chemistry development using photocatalysis, electrochemistry, as well as recently some biocatalysis in flow also are reported. And next we go to photocatalysis in flow. What is the advantage if you take photocatalysis reaction in flow is there is better control reactions and the reaction time is short, and moreover it is scalable. So when we do develop reactions in the flow small scale, but however in the scale up in the big reactors it is very difficult to maintain get a very efficient reaction condition in the scalable. However, in the flow reactor, we can easily do scalability because the reaction happens in a small micro reactors. So it is scalable, it is already proven concept. So obviously it is a sustainable chemistry. So far, what are the give background photocatalysis in flow has been applied, specifically drug intermediates. These are some literature classic examples. Brivaracetam, fentanyl hydrochloride, the rosuvastatin is a blockbuster drug for the lipid lowering agent, and also including some rearrangement reactions we can see for synthesis of ibuprofen. It has been successfully done in the photoflow reactions. The same way it has been applied for the synthesis of artemisinin. It is used for the anti-malarial drug and also nutraceuticals, vitamin D. It has been successfully applied the photocatalytic reactions in the flow reactors. So this has been done already in the gram scale. So it is signify that it can be scalable and used for the manufacturing of the pharmaceutical products. And now we go to our lab. What kind of work we are doing? This synthesis. The first line you see the synthesis. We have finished from small to big molecules. We started with small intermediates. We can see this is used for the certain series of drugs, and though it is not flow, but just we have done it that I am showing the slides. Out of this 6 flow we have applied, this is the tavaborole and this crisaborole, and in the synthesis of trabectedin we have already used as one of the intermediate by flow chemistry using photocatalyst we have used. And also some of the steps of this daclatasvir, it is antiviral drug. We have used one of the 2 steps out of this in the flow reactor in the flow chemistry. So today we'll discuss about this borylation in photocatalysis and also one methodology we have developed using flow reactor. These 2 we'll discuss. This is the first methodology part. What we want to see is the selenochemistry. Our lab working on different seleno compounds. More of seleno compounds, the significance is they are used recently applying several papers for the neurological, especially the Alzheimer's disease. And also these are precursors for native chemical ligations. You'll find many papers on the NCL native chemical ligation using selenoesters. And also these are synthones for the selenoacids and acyl radical synthone. So it has from chemistry point as also biology, it has the significance. So we develop a new method using flow reactor for this selenoesters. So this is the reaction. We develop selenoester directly from carboxylic acid. It is a 3-component reaction. One is carboxylic acid. One is the source of selenourea, which is source of selenium. And third one is a Michael acceptor. The classical method, carboxylic acid is converted to acyl chloride, and then it reacts with diselenides in presence of metals to get the selenoester. Here is a 3-component reaction, this carboxylic acid and Michael acceptor and the selenourea, which act as a, we have given heading, it is a bifunctional because it is a source of selenium as well as it acts as a activator for the, it's like double hydrogen donation to the oxygen of the Michael acceptor. So it activates the carbonyl group and it enhances the reactivity of the seleno reagent. So dual role it is playing here. So we get it, we have done the traditional batch process as well as we have done in the continuous flow process. So in continuous flow process, it is 120 degree centigrade with 30-minute residence time. We have obtained the desired product. This was published recently in the ChemCom. This is a cover picture, back cover picture of this article. So the same way in the reaction happens in basic medium. So we design new seleno reagents. We can act as a seleno reagent as well as it contains some basic moiety. You can see diethylamine or dimethylamine or a pyridine system. So it will act as a base also. So it will have seleno source. It will act as a activator as well as also base. We don't have to use any base. So we designed and synthesized this third middle row series. We have synthesized the 3. So we get the best for this compound 7G. We can act as a base also. We don't require any base for the reaction. So it will act a base and also source of selenium as well as the activator. So the first part is published. The second part with some more extension we are supposed to publish. And this is the mechanism. The selenourea, it reacts first with the Michael acceptor. It undergoes 1,4 Michael addition to get the intermediate 10. Then carboxylic acid reacts with this imine, and finally, this nitrogen gives back the electron, and this carboxylic, the seleno part will knock out and go out, and finally it reacts back or attack back to the active ester to get the selenoester finally. And byproduct will be your selenourea. This compound 16 will be the byproduct. And we have isolated this compound 10 to give the evidence of the reaction mechanism. And the main side product is compound 9. You can see the diselenides. And we control this reaction. In traditional process, it was higher. We are getting at least 50% of diselenide product. However, when you use the flow reactor, so we are able to control this. We get the desired compound higher, and this impurity 9 was reduced. So that is a help. We can flow reactor can help in reducing the impurity it was observed. So now we are changing this Michael acceptor, and we'll publish this in the next few months. So Michael acceptor is being changed with different halo compounds, and also we are taking different carbohydrates as an anomeric center. How can add the seleno to the anomeric carbon of the carbohydrates, and also different halo compounds. That will be the next follow-up. And next we go to this photocatalyzed borylation reaction. So the background is why boron compounds as a pharmaceuticals, though it is used since long back, the boric acid, but recent these 4 drugs you will see, the tavaborole and kesaborole, these are antifungal used by for the treatment of foot and nail fungal infections. And this bortezomib and ixazomib, these are multiple myeloma or this type of cancer disease it used. These are very highly potent and high cost anti-cancer drugs. So this is the background of boron in the pharmaceuticals. So our objective was to synthesize boron compounds from a halo compound. So this is the background what is so far has been done. It has been done by the organocatalysis in the JAX 2019 and also directly without any catalyst but it is with UV 254 nm. So it is UV region it has been done, and also by metal catalyst like iridium and nickel has been reported. JAX 18 and 20 it has been reported. So what is difference is ours is we do the oxoborols, the toaborol and crisaborol contains the oxoborols particularly. So these oxoborols we synthesis in single step. Earlier it was in 2 to 3 steps. You can see this, you take the orthobromoaldehyde, reduce it and borylation followed by the this pinacolate, deprotection and cyclization in acidic medium. So other one is same way, first reduce the aldehyde, protect it and do the borylation by the lithiation reaction and the followed by cyclization. So we propose here the single step, the borylation, reduction of aldehyde and the in situ, we can say telescopic process, in situ cyclize it to get the oxoborol. So this is novelty here is one is first borylation obviously reported by organocatalysis, but we here observed that the bispinacolate diboron in presence of basic medium at 410 nm, it is reducing the aldehyde also. So in use in this case 2 moles of bispinacolate diboron, so that will also reduce the aldehyde. It will also facilitate borylation. Then in situ we deprotect and cyclize to get the tioborol. So this is just one example we have published and filed the patent. Now we are screening out different organocatalysts for this aldehyde reduction in presence of B2PIN2 and also only 254 nm whether it can reduce the aldehyde and do it. So that will the follow up with other examples. And this is the mechanism. First part is this we have used in this case for phenylpyridine. So this is the organophotocatalyst. So it reacts with diboron to make this radical complex 9, and it is also there is possibility of formation of phenylpyridine can coordinate with boron make the complex, and there is possibility it is also feasible. So next is this compound pyridine boron complex 9 radical. It can transfer. It can be acting as a super electron donors, we can say. So this activate the halo compounds by single electron transfer. This is the classical mechanism to generate the radical intermediate 11, and then it further reacts with the diboron to get the boron complex 12. And now novelty here it is. Here is also this aryl radical intermediate 11 reacts with 4-phenylpyridine complex 9. You can see this radical complex to form the 12. In this case, what happens? The single electron transfer, reduction of formyl group by this super electron donor 9 also happens. So a single electron transfer also happened to the aldehydes from this complex 9. That is the novelty. So it can also reduce the aldehyde to get the alcohol. In situ, we cyclize it to get the boron complex. So this is one example we have done. Now we have screened some more examples of this different photocatalyst, and also without any photocatalyst 254, we have some results on this that will publish it or will file the patent. Yeah, that's all. So these 2 examples, though we have more data, so we highlight here 2 examples only in this case. And yeah, these 2, that's all. This is short presentation from my side. Thank you. And this is our institute photographs, the department, and these are the postdocs, 2 postdocs and 4 PhD students work in this flow reactor project. And this is Department of Science and Technology, Department of Pharmaceutical, Government of India, the funding we have received for this project. Thank you. Thank you all.
Thank you, Dr. Swan, for your nice presentation on a flow reactor for pharmaceutical production. So there are a few questions in our chat. The first question is from Vinod, how this photo edgy lamp helps in reaction catalysis?
How this photo lamp?
Photo mercury lamp helps in reaction catalysis.
This is that I told that for boron, you can see this by photocatalysis. It will break the diboron bond. It will generate the boron radical. Other way also, that will also, if you take halo, it will also carbon and halogen bond can also break down to make the radical. So it proceeds generation of first radical, then it will go by the further radical reaction, formation of this carbon and boron bond formation. The first step is it will help in facilitate in the radical generation and breaking of the boron-boron bond in bisphenolate diboron.
The second question is from Thieme 1679. So they are asking like, great to see the synthesis of tevaborel in single step. Is it possible to perform it the reaction without flow chemistry setup?
Yes, without chemistry also flow chemistry we can do it. That is, we have tried also both way. First we did traditional, followed by the flow chemistry. So as I said in the first, flow chemistry helps in scale up, we can say, and uniformity. And some cases we can see the impurity formation chance is less because once your product is formed, so that goes out. It is continuously flowing in small scale. In the batch process, though your some product is formed, still reaction is not complete. Some starting is there. So further your product is also getting exposed to your catalyst or further reaction condition that also undergoes further reaction for impurities. So in flow, what happens, the reactor, we take 5 ml or 10 ml reactor. So after 10 ml, it is pushed out, again fresh 10 ml comes in. So your product is formed, it goes out, it is not exposed further to the your reaction condition or catalyst. So that helps in the less impurity. And flow reactor, obviously, it helps in scale up photo. That's why many years before photo has been started, but still scale up has been issued. You'll find some companies have started the photocatalysis scale up. Though in India, it is still in the lab scale. They are now in the process of maybe they will go very soon for the big scale up. But we see European countries, some companies have already started manufacturing by photocatalysis using flow reactor.
Thank you, sir. So the last question is from Bihu. So she is asking, if we use selenourea, do we need the absolute dry conditions or not for the replacement of chlorine with selenium in the substrate?
We have used so far dry condition because sometimes we are using base. So if we use water, the selenourea, and heat it, there is possibility of breaking down of this that intermediate complex. So it may interfere the carboxylic acid and water, they may compete. We may not get the ester, we may get the hydrolyzed product. So obviously we have to do in dry condition, especially for this reaction. As well as there should not be any oxygen because the dimer forming seleno will dimerize in presence of oxygen as an oxidant. So both water and oxygen should be avoided.
Thank you for your reply, sir. Professor Laha, over to you.
Yeah, Dr. Swain, again, it's a very nice work and a very nice presentation. I don't know, we are already behind the schedule. Can I ask one question quickly?
Yeah, sure. Oh, okay. You ask.
Yeah, so I just one quick question. Then what is the role of the compound 10 in the mechanism that you have shown? Your compound 9 is the super electron donor. So what is the role of compound 10? Is it also involved in the reaction, or what is the role?
So it is, we show this because there is possibility of formation, but mostly that is our prediction. Compound 9 is enough to facilitate further reaction of like reduction of aldehyde by single electron transfer. But theoretically, there is possibility of formation of 10. So we show this. There is possibility.
Okay, all right. Remaining questions I will ask over phone. Okay, thank you so much.
Thank you.
Can we resume to the
yes, Professor Laha. Actually, I'm sorry for that technical error. Yes.
No problem. So we could start from the presentation where we left.
Yes, yes, I'll do that. Is it visible in my presentation?
Yes.
Yeah, thank you. Thank you. So I left here. Actually, I'm talking about this pentacyclic core. So we had initial strategy making this pentacyclic core from the tetracyclic core, but when we had some yield issues, then we had to revise this strategy basically from this ICR. So where first we have pre-functionalized, and then it was subjected to this Victor-Spengler reaction conditions, that is TFA at 0 or lower temperature. So this reaction was very interesting. In this, multiple reactions were taking place. So in this case, actually acetal deproduction, bark deproduction, and then formation of this imine, and then involved in the cyclation. So in this case, actually, there is a one CC bond and a CN bond with complete stereo control. So after accessing this pentacyclic core, we could easily convert pentacyclic core to this macrolin and elastomycin natural products, and elastophonidin A and elastophonidin D, elastophonidin, and presently we are towards this completion of macrodacin. So not only that, just a small change from N-methyl to N-benzyl. Actually, this was a very not an easy reaction to introduce this ethyl, but it was actually required a lot of optimization to synthesize this compound. Then after that, we could make some advanced intermediates which were already reported, and we claimed it as a formal synthesis of this molecule. So after achieving this one, so we could make several of this advanced intermediate, and which were already used in this macrol indole alkaloids, and including one product synthesis also we have recently completed. And coming to our research areas, as I said at the beginning, multiple research areas, but today topic was this process for APIs. So in this, actually, we have worked on several natural products. If you see drug or any medication composed of two components, so the first one is actually a very important component, and the second one is known as a excipient, which is an inactive substance, but helps to the drug as a vehicle. And so these APIs are generally manufactured through a variety of processes that include chemical synthesis, fermentation process, or recombinant isolation from or recovery from a natural sources. So a combination of you see, taking from the milligram scale to the manufacturing scale is the role of process chemist. So the goal of process chemist is to develop synthetic routes that are safe, cost-effective, environmental friendly, and efficient. So once if the manufacturing setup is established, so completely changing the process is not viable because several potential impurities in the synthetic route and supply chain concerns and raw material sourcing. So without affecting these points, if any process improved, so most welcome in the pharmaceutical industries. So with this, actually, we have worked in various APIs. In some of the cases, it was key starting materials. So I'm going to briefly show in today synthesis of this dapagliflozin, advanced intermediate, and ledipasvir. So coming to this ledipasvir, Gilead Sciences has developed for the treatment of hepatitis C virus, which is a leading cause for jaundice and hepatocellular carcinoma and liver transplant in a SCV infected patients. So it is used as a in combination with the sofosbuvir under the brand name Harvoni, which is a direct acting antiviral agent. So ledipasvir inhibits viral important phosphoprotein, that is NS5A, which is involved in actually replication, assembly, and secretion. So at the beginning, the price of this ledipasvir was very high. If it is a 12 to 24 weeks treatment cycle, it is costing very heavily. But this Gilead Sciences justified that outweighing the cost benefit when actually if you look at in the liver transplant or the partial treatment. So if you see the synthesis of this ledipasvir, there are mainly 3 fragments, and the 4th fragment basically it is nothing but just a mark valin. So after deproduction of this bark, so introduced this mark valin at both the ends. So out of these 3 fragments, where the fragment 1 and fragment 2 are less complex, and the fragment 3 is highly challenging. And the synthesis, if you see the key cycloaddition reaction is actually with excellent selectivity and yield, and all other steps are mostly straightforward. So we could not see much scope to improve. But when it comes to synthesis of fragment 1, it is started from the 2-bromofluorine, where initially introducing iodine, then difluorination here. In the presence of isopropyl magnesium chloride, it is reacted with this vendrabomide to get the fragment 1. So alternatively, it is also synthesized to first Friedel-Crafts reaction, but in this there is an active keto functionality which is required masking with diacetyl, and then the difluorination is done. But the shorter routes, either just making up a difluorination with the low yield or even further Friedel-Crafts reaction is didn't work. And the fragment 2 synthesis actually started from the diol, which is again synthesized from the dimethyl malonate treating with the 1,2-dibromoethane. So this diol is converted to diiodo compound and then reacted with this glycine to get the proline, bicyclic proline derivative. So finally, a simple enzymatic resolution provided the fragment 1, but in this case, the enzymatic loading was very high, 30 to 40%, and often only 50% or less than 50% of the yield, and same with the even chiral resolving agent. So there is an alternative route for this, starting from the 4-hydroxyalproline, so where there are a few steps, but this was again a challenging converting this volute into the cyclopropionation. So in this case, during the cyclopropionation here, this free carboxylic acid also getting ester, and not only that, even the unreacted starting material also becoming the ester. So at this stage, these two are inseparable even after hydrolysis. So both again inseparable mixture. So to remove this unreacted starting material, actually iodolactone is carried out as an additional step. So alternatively, this is done using the bromo as a cyclopropionation, and then removing this halogens using the palladium carbon hydrogenation. So in short, if you see the fragment 1 synthesis and fragment 2 the synthesis here, after synthesis of both the fragments actually converted using this keto functional alpha chloro keto functionality and acid converted to the imidazole. So at this stage, we consider in case like if we can late stage a cyclopropionation and this difluorination, we can avoid a protection of this keto functionality as well as unwanted transformation of this acid becoming a ester as well as unwanted this hydrolysis. So with this idea, we thought like we have used this known starting material which are already used in the process to converting this imidazole, so which is actually part of the molecule. So with this advanced intermediate, we have attempted for the first cyclopropionation here, and it was almost good to excellent yield. And then we carried out this difluorination again without an issue. We could get this advanced intermediate, and then just to show the advanced intermediate, we have also coupled fragment 3, and finally the production of this bark protecting groups. Then we could achieve this lead positive. So in a short, actually we have not changed the beginning raw material or reagents. So we have only changed the order. So in that we could reduce the number of steps. And another advantage in this is actually the NFSI reagent. If it is at the very beginning, it is required in a huge quantity, but where it is in the late stage, the quantity of the NFSI is drastically reduced. So with this lead phosphate, we have actually published a synthesis and filed a patent. And in a very similar, we have made another advanced intermediate of this gliplogens. So you know that diabetes is a prevalent and growing concern around the globe, and there are majorly type 1 and type 2 diabetes. I'm not going in detail, but you can see that way back from 1915 there are several advancements based on the targets. So the recent one is actually gliplogens, SGLT2 inhibitors. You can see the structure. Some of them are best selling in the market. So the major task was actually this diaryl methane fragment in this dapagliplogen, and there are other gliplogens. You can see varying only the one of the aryl position or substitutions. So if you see the synthesis of this diaryl methane actually started from this benzoic acid converting to the acyl chloride and doing this Friedel-Crafts reaction to get the ketone and then reduced here. So when we have seen the backward integration for this particular compound, it is in the literature procedures showing that it is synthesized either from this 4-step synthesis or 1,4-diallo-benzene using orthochlorolithiation here, and then carbon dioxide was used as a electrophile. So we consider instead of carbon dioxide as a electrophile, if we can use this ethoxybenzoaldehyde or any aldehyde going to act as a electrophile, and then this alcohol can be easily reduced following the very similar procedure developed earlier. But advantage with this is the number of steps are going to be only 2. And another advantage, it doesn't require any column purification. So easily we could crystallize this compound. It is not limited to this method or protocol to this dapagliplogen. We also apply to synthesis of this empagliplogen and impragliplogen. So this is we have already filed a patent, and so these are the small changes. Actually, with just changing the route, we have actually cut down the number of steps and then improved the overall yield. And in this case, as I mentioned, when you change the process, sometimes there would be this raw material sourcing concerns. But in this case, so again we have used the same raw materials, and most of the time even we have not changed the reagents. So with this small presentation about this active pharmaceutical ingredients. So actually this is done by hardworking students past and present, and this natural product synthesis mostly done by the PhD students, but this API's process development mostly done by this Ramu, and again he is registered for a PhD and going to complete soon. So I thank all who are participating in this seminar and for your patient listening, and I'd be happy to take any questions if you have. Thank you very much. Professor Laha?
Okay, yeah, I mean, Rohit, there are a couple of questions from the audience.
Okay.
Let me see. Okay. Okay, no, I'm sorry, no, there is no question for you. But then
okay.
I got a question.
Please.
So in the synthesis of the first drug, lady
phosphate.
Right? So what was the overall yield immune synthesis?
So and it is yes, actually it became close to or slightly more than 20%, whereas it was a little less in the inventor's route. It is coming around 15 to 16%, but it is not a very much difference, but overall it become more than a 20% now. But if I see the overall, the number of steps actually could reduce, and then some of the reactions, if you see the cyclopropionation and then difluorination at the late stage, so without a problem we could carry out these reactions. So there is a some handling. It became a little easy to handle because of these changes.
Okay, so yeah, I mean, so what was the scale did you make the lady phosphate? How much quantity did you make?
So it is not, yes, actually in the laboratory scale we have done mostly in a few reactions in a gram scale, but the final steps we have done in 50 milligram scale or 100 milligram scale. So at that time, like we were trying to patent, and then of course after that we have published. But yeah, if there is any requirement and if any approach from the industry, we thought of like doing the more scale. But as of now, it is done only the laboratory scale, mostly gram scale. Yeah.
Okay, all right. So in the meantime, we got one question from the chat box. So the question is,
okay,
was there any lithium halogen exchange product formed in the reaction with lithium and carbon dioxide? Was there?
No, we have not seen any lithium about the fluorination step, or is that clear?
Yeah, in the reaction with lithium and carbon dioxide.
Oh, okay. So that is probably in the second part. Yes. Yes.
Yes, in the last slide. Yes.
Yeah, yes. So here we have used different bases, but we have used LEA to remove the proton, just ortho to chloro selectively. So there is a literature support for that based on that we have taken actually. So when we have used in place of chloro, if we use a fluoro and chloro, but selectively we could do the ortholithiation at this ortho position of chloro or fluoro, not at the bromo. Then we have added electrophile like benzaldehyde to get the product. Did I answer your question?
Well, it's not a question from me, it's from the audience.
Okay.
I think that the audience is happy, so far I don't see any other question.
Okay.
yeah, so I think there are no more questions. So I would like to thank you for the excellent talk on time. Thank you very much. So
Thank you very much.
So before I hand over to Rohit, I just wanted to say a few words. I really want to thank all the speakers, the 4 speakers today, for their excellent talk, the inside out, very quite informative. And then also I thank the participants who attended the today's event. And then also would like to thank the contributors to the special issue in synthesis, and that's what this seminar is linked to the special issue. And finally, I'd like to thank the Chemistry Journal's team, all the members, especially Rohit, Fadwa, and you, and several others who are in touch with us for several weeks, have been shooting us emails for many times. Thank you so much for organizing this wonderful event. Now I hand over to Rohit for the closing remarks.
Thank you, Professor Laha, and thank you all these speakers for participating in this event and making it successful. Thank you all the participants who attended this event. You will be receiving recording of this exciting webinar after this closing of this seminar. And also I'd like to invite you all for our upcoming next webinar, which is will be about the best poster prize for synthesis and synlet. This will be the talk by Professor Shantanu Mukherjee and Professor Sato. This will be next month. I encourage you to kindly register for it and join us in this exciting webinar also. Thank you all of you for joining once again. Thank you all the speakers once again. Thank you Cassini for their all the technical help, and all my team, and most probably Padwa, for all the help in organizing this exciting one. Thank you very much.
Should I take off? Go down now?
Yes, we can take so. Thank you very much.
All right. Have a good day, everyone.
Thank you. Okay, please.